Hello, everybody. My name is Emma Lejeune, and I'm an assistant professor at Boston University. And I'm thrilled to be hosting the Closer Look Journal Club with my colleagues Manuel Rausch at UT Austin, Matt Bursey at WashU, Adrienne Buganza-Topole at Purdue, and Johannes Weikenmeier at Stevens Institute of Technology. The group of us kind of started this journal club just sort of as a means of connection during the pandemic, basically just really as a simple way to bring some high quality speakers to our own labs and our own graduate students. And then we decided to open it up to the broader research community, which has been a really cool experience so far. We've had people kind of from all over the world who show up virtually in these meetings, which has been a lot of fun. So with this sort of season or semester of talks, we really want to encourage the audience to speak up with questions. So we've asked our speakers to only prepare 20 to 25-minute talks so that we can have a long Q&A and discussion session up to maybe 20 minutes or so at the end. So please keep this in mind as you listen to the talks, because there will be plenty of opportunities to ask questions. And as all of you know, we have a really amazing lineup of speakers. Our first speaker is Professor John Estrada from the University of Michigan. John has a bachelor's degree in material science and engineering from MIT, and he completed his PhD at Brown University in solid mechanics in 2017. And he's been at the University of Michigan for the past couple of years, where he runs an experimental soft mechanics laboratory. He'll be presenting his recent preprint titled Neural Cell Injury Pathology Due to High Rate Mechanical Loading. And I'm really excited to hear what he has to say. So John, please go ahead.
Thanks, Emma. I appreciate the kind introduction, and thank all of you guys for hosting me today. It's a real privilege and honor to be the kickoff for what's going to be a really awesome season of Closer Look. So thank you. So my group generally does experimental research in mechanics of soft and biological systems. I'll talk maybe a bit more about that later, and actually if there's time, I might give a virtual tour of my lab on GatherTown. At least I can screen share it. But today I'll be highlighting a study that in one sense I'm finishing up with Christian Frank, my PhD advisor, formerly at Brown University and now at UW-Madison. But in another, it's really a launching pad for answering a whole host of new questions and methods of quantifying very high rate injury, and has just been really firmly advanced by my great friend and colleague, little brother in science, Harry Kramer. He's here on the call, I think, and I'd love him to take part in the Q&A session with more A's than Q's, I hope. But I would be remiss in not mentioning that he's headed to the job market and is just fantastic. Harry's the best. I'll also mention up front that this work was primarily funded by Tim Bentley of Code 34 at the Office of Naval Research. All right, so let me see which way I need to, there we go, I can click on that. All right, so I want to start with asking two questions here, which are really, what is TBI, what is traumatic brain injury, and why we should apply the lens of mechanics to study it. So TBI can result from either direct or indirect loading to the brain, and it negatively alters brain function in some way. Why do we study it? It's a common disease. It affects 1.7 million Americans annually. It's the leading cause of death for people under 45, adding up to about 60 billion in healthcare costs annually. And there are a whole host of different mechanisms of damage that range from blunt trauma and closed injury, such as shaken baby syndrome, to high-speed penetrating ballistic, crushing injury and crashes, pressure waves from blast injury, and now more commonly, directed energy, such as in, you may have heard of Havana syndrome. In the field, however, the severity of these injuries is commonly assessed on this sort of general 1 to 15 scale of injury called the Glasgow Coma Scale by paramedics. And, in one sense, and I know I'm speaking to the choir here in that some of you do brain-related research, TBI is sort of the ultimate inverse problem. It really just isn't feasible to back out, what kind of loading events happen by seeing a patient present with trauma or concussion-like symptoms. I myself have had 3 concussions in my life, my last being in college, and a big source of frustration for me at the time was what happened, because both I didn't remember the event, and that even if I did, there was no way to translate the mechanics of what occurred to any kind of damage my poor, remaining neural cells experienced. Thus, we really need urgently injury criteria. This is something we're really striving towards. So let's take a couple of the TBI modes from the last slide and roughly cast them in terms of speeds here. All the way on the left, we have a range of tissue strain rates which occur in the normal day-to-day sloshing around of your brain. You walk around, it sort of moves around. Don't think too much about that, but we can walk around without damaging our brains because, for one, they're suspended in our skulls, which act sort of like a viscoelastic mechanical damper. but also because the strain magnitudes and rates are comparatively low. Now, as mechanical events begin to become more injurious, it's usually the increase in rate that actually starts playing a major factor here, seen as, for example, boxers and football players exhibiting abnormal broken tau proteins, which is sort of like a protein that holds microtubules together, and when it ruptures, it signifies a different kind of damage. But these are seen in sort of post-mortem images. Now, as the rates get even higher, such as in blast and directed energy, we observe injury, but it's harder to get a read on what exactly goes wrong due to, again, because the mechanics problem is complicated. So even in the realm of conventional material testing, high rates aren't easy to achieve. I mean, you've probably seen uniaxial load frames like Instrons and MTS systems. These can reproduce low rate, low stretch, but we need more exotic systems like the split Hopkinson or Kolsky bar setup, or BLAST's shock tube platforms to conventionally get access to these faster rates. And even more commonly in biological systems, you don't have enough sample to even test in these kind of setups. So this study was especially aimed towards understanding the ultra-fast regime, which is what distinguishes BLAST and directed energy TBI. And so to give you a sense of the speeds we're talking about here, a typical blast wave over here on the right starts with a shock front, which has the highest strain rate and pressure amplitude. Shock only lasts for a couple microseconds, whereas the full blast wave is sort of hundreds of milliseconds. Peak pressures are pretty high. We're talking like 2 to 3 orders of magnitude above atmospheric. And a lot of other scientists have looked at the overpressure mechanism, which is like apply a shock tube, make a shock front, and see just an overpressure create damage. And there's sort of mixed reviews about that. It's not entirely clear that there's a direct correspondence of more pressure means more damage. But on the other hand, there's this tensile portion of the wave that when that actually passes through something that's incompressible like water, there's a separate mechanism here, which is bubble nucleation. And it's unstable, it grows, collapses, and then you have this what's called an inertial cavitation mechanism that can create its own kind of damage. So this is something you want to keep track of. And then these directed energy systems, which include directed ultrasound, high energy ultrasound, and lasers, can create cavitation even directly without inducing shockwaves specifically. So, we need to sort of characterize this. And we've seen high rate inertial cavitation before harnessed clinically. And for those of you who don't know what inertial cavitation is, if you've ever seen the party trick of if you have like a beer bottle half full of water and you whack it really hard on the top, the reason it breaks is actually, and you can sort of see in the video over here, the reason it breaks is due to cavitation. You get these bubbles to form, and then the plug of water smashes back down, blowing up the bottle. It's the same exact reason. If you drop a test tube on the ground, it shatters. There's actually a very cool PNAS paper about that from a few years ago. It's also the same phenomenon that causes knuckles to crack and the erosion of this pump impeller down here on the bottom left. But it's actually deliberately used as a surgical technique for breaking up tissue or blood clots, occluding vessels, kidney stones, and more. But generally, historically, the focus hasn't been on the mechanistic link between cavitation dynamics and cell injury. So that's a thing we have to quantify to move forward. So that's our goal. We're going to try and connect injury of cells with the inertial cavitation dynamics process here. So we sort of hypothesized that cavitation is an important shared mechanism of cell damage here in these two TBI classes. So schematically, this is the idea that our directed energy or impact, whatever kind of fast load event we have incident on the brain here, produces a stress field. And the stress field is going to have two different components. You can break up a stress field into a deviatoric part and your hydrostatic part. And so as the brain contains a large amount of incompressible water, you can imagine that as a compressive wave goes into the brain, it'll rattle around, and then once it hits a surface, it'll flip and go tensile. And that pressure drop is what's expected to nucleate these unstable bubbles that expand, run out of energy, and collapse back down, harming tissue in the process. And that harming of tissue is what we really want to characterize, this primary injury phenomenon, which is the mechanics part. This is the purely mechanical aspect. So, did we exceed our failure strain or stress? Now, of course, that's not the only injury metric. We could look at secondary injury pathways as well, which is this sort of biological or the bio catching up to the mechanics here. So instead of just broken versus non-broken, this would be like time evolution. And this would include your sort of biochemical cascades, such as apoptotic or necrotic cell death pathways, right? But we're trying to contribute data points here. We're trying to contribute with a certain amount of, and over here on the bottom, we've got our strain rate and our y-axis here is our strain value. And there's a combination here that produces damage at low strains. Even if you move things very fast, you can break them viscoelastically, and you can actually move things quite a bit. But if you do so slowly, they won't break either. So we're looking at the combination. And I do apologize. I'm not exactly monitoring the chat, but I do see that there was a question. And the question was, when I'm talking about injury, I'm focused on low frequency, but high impact. It's actually really both high frequency and high deformation here that we're looking at. And the question of who is Steve? Good question. It's a skeletal friend of the skeleton we saw so far. Great question, man. All right. So here we go. So now knowing the length scales in the brain, or given some length scales in the brain, we want to try and focus on the length and time scales that are important, right? This is a classic sort of mechanics problem, right? So we want to connect all the dots here and find the size scales that we're really dealing with. And so we looked at sort of post-mortem studies. There's a big one out of the Boston University group. And so what we sort of see is the damage scales of interest relating to blasts seem to be on the order of sub-millimeter, but larger than cells. So it's sort of in this middle ground of many cells, but not quite very large tissue. And we want to connect this to what we can, look at and observe in terms of the inertial cavitation problem. So, the difference here, though, is that we want to look at the actual event happening. We don't want to just look at postmortem and this is the result, right? We want to see the immediate, before and after response, but also the during motion of this bubble that's going to be creating the damage. So that's what we really want to get to make something with the entire history that we can track. And then we could look at this further and say, well, on the single cell level, it's important to know what's breaking or what structure gives way to deformation. I'll talk more about this later, but we have to start even with like, is our continuum assumption okay? And it seems like it sort of is, because we've got this fibrous matrix that well encapsulates our cells. And because we move everything very, very fast, we're going to assume a viscoelastic continuum for the surrounding, but it's really because we move this really fast that the fluid doesn't have time to flow with respect to the matrix. So it's sort of a fluid-filled gel that moves very quickly. So it seems to be an okay assumption, but it's certainly something we can look more into. All right, so now I want to give you guys a sort of basic version and overview of what our experimental process was. And so here's the overall depiction, which has 4 image acquisition states, which I'll describe subsequently in more detail. Just want to give the quick version, the TLDR version first. So we start by fabricating and growing our neural cell cultures. We stain them for calcium AM, which is like an intracellular esterase activity measurement. It's esterase activity is a normal metabolic process. It's in all neural cell types, so we can really see everything that's inside our cultures. Then we take a coarse image stack using confocal or multiphoton microscopy, Z-stack, so we're looking at 3D volumes for a pre-injury reference. I need to click more as I go through these slides. Then we're going to induce injury. So we then, after the duration of that, that volume, we then hit this with a laser pulse, which is going to create a bubble locally. And so this is sort of an old video from a prior paper that I had, but essentially we're growing a bubble, it's expanding, collapsing back down, and oscillating. And we're looking at that radial dynamics here. Then we measure the dynamics. So I guess we measure that with an ultra high-speed camera, and then we're going to measure the post dynamics volume, again with multiphoton or confocal microscopy. And after that, we usually do some sort of endpoint imaging, and that'll be with applying aldehyde fixation per row, and later, stain and image volumes for structural components. So that's sort of the general gist of it, but these rates are comparable to what we see in actual blasts. So, so far, so good. So I'm going to replace those four data set things with this little procedure schematic, and that'll be there as we go through the next couple slides. So here's the more crunchy version of what we do. First, all of these cells are actually primary dissociated cells from rat cortices. So we take the cells out, we mix them with collagen 1, which is not in the brain, but it's a pretty good scaffold for cells. More on the choice of collagen, we could discuss that later. But anyway, we cast these, incubate them for 20 minutes, 37C, put cell media in, we let them grow over about a week, and then they reform these networks and look really quite nice in like a galaxy 3D kind of perspective, but also they're very well connected and healthy. And then we sort of take those so that would be our initial reference. And then we do this damage, right? So we fire in a laser pulse through our microscope, which we had to fit together. And we're going to backlight this with bright field imaging, measure with an ultra high speed camera. And like I said, there's sort of a bubble that's generated right at the focal point of this objective. The cool thing about this here is that we're actually looking at the same plane we generate cavitation in. So everything's nice and collinear. So this is like a nicer video, a more recent one of the expansion, collapse, oscillation process of our bubble inside our gel. And so this is sort of a sideways view of this where we have a reference, we do our deformation process, and then we have this damaged version afterwards, which is, we want to quantify the difference between those later. From here, we get to extract radii over time, which sets a couple new length scales in our mechanics problem. Again, more on this later. But this sort of defines what our total stretch we apply from a bubble is to our material. So these bubble dynamics really define our deformation field during cavitation. From here, we want to then look at now, all right, we've looked before, we've looked during, what happens after? And here's a maximum intensity projection of this post-cav damage. And I want you to notice a few different things with the reminder that calcium AM is a measure of internal metabolic activity. So first, cells close to this cavitation epicenter look comparatively worse off. Sort of to be expected, right? I mean, they're nearer the ground zero here. And it's plausibly due to stretch or strain. It seems, there's a radial drop off to it. There additionally seem to be different injury morphologies. So if you look at different cells at different locations, some of them are sort of morphologically normal. These look basically healthy to a trained eye. And then as you get closer to this epicenter, there are sort of funky things that happen with the little projections here. These are sort of blebbed. Ones that are closer sort of have their arms torn off. And then finally, ones that are really close are completely fragmented entirely. So for the purposes of this study, we'll treat damage as fully broken or severed projections. Really, we're going to be looking more at something like this. But the definition of damage is really up to the user here, given you have this system to resolve your damage metric. Like we could, in theory, look at projection blebbing as the damage metric, but we're choosing to sort of look at this blunting. Now, I've gotten this question a lot of, well, is it thermal damage doing this potentially? And interestingly, it wasn't a thermal effect, at least as far as we could tell, because as cavitation generation is sort of a stochastic process, sometimes the laser would fire but wouldn't nucleate a bubble. And in those instances, there wasn't really any appreciable cell damage. So we really think this is a mechanics phenomenon because when we would grow these bubbles, we really saw resulting cell breakage. So, now we have some new length scale, right? So we've got some length scale that corresponds to projection damage. And we have our before length scales from our high-speed images. So we have to sort of connect them all. And the question is really what we do with them. So we use two customized image processing procedures to assess damage length scales. Again, based on our choice of metric being the sort of arms torn off, right? Which is that projection fragmentation. And so for the first procedure, we essentially take image differences. We have our before, we have our after, essentially take the difference of those two, which is shown here in a purple-green anaglyph image. If you've ever seen red-blue images, you can put on 3D glasses and sort of see it that way. And then add in cleaning them up, binning them into radii, and we can establish a radius value per experiment corresponding to a set percentage of the total loss signal between pre and post imaging. For endpoint imaging, we don't have the luxury of before images, so we have to do something different. And and that's generous, I should really say, Harry manually tagged like hundreds of fragmented projections and channels for F-actin, which is in all neural cells, beta-3 tubulin, which are microtubules specific to neurons, and MAP2, which is found in neuronal dendrites specifically. And each one of these locations is then tagged in the data set, accounting for also any 3D difference to the center point, and establishes a radial value, again, with more scaling here. So then that's sort of the end of our data acquisition, and now the processing part happens. So I like to refer to this as a tale of several length scales, because it's a question of what we do with all of them, right? We've got like a zillion of them here. And so we sort of look at them overlaid on each other and compare with respect to one length scale what happens at a different one. And this actually works pretty well and establishes actually a sort of probability curve way of dealing with, well, at what stretch can we reliably say cells were damaged kind of statement, right? So first, again, we need to assemble all our experiments into mechanical quantities. But like I said, comparing radii to each other is actually pretty useful, and it non-dimensionalizes as stretch or strain, right? Radius over radius usually gives you something like stretch. So it's pretty natural mechanically at least, but we need to know first what our loading conditions are. And so if we compare all of our maximum bubble sizes to the sort of remnant bubble that's left over after damage occurs, which is what we call our equilibrium bubble size, it's actually pretty freaking linear. This was actually striking to us. I mean, I was not expecting this kind of linearity, but we really had distinct linearity, which actually tells us essentially that there's a set amount of stretch that we apply. It's almost like we've made a stretch condition for our deformation field, and this system sort of naturally adheres to that, which is sort of crazy town. And it was, there's a value to it. So 7.4 is the R max over R equilibrium for this entire thing. And that's maximum size of the bubble to equilibrium size. And even better, it sort of looks like that this image processing derived primary injury radius, where this magenta one is this one single point over here, and we have 200 something tests here. This seems also pretty linear, suggesting that strain to failure is actually pretty consistent too, which is really interesting. So now we just need to sort of connect, well, what did cells experience at this location in space with respect to Rmax? And we're good to go to sort of find a cell failure criteria. So we can do that. We first have to assume something about the deformation field, or we could alternately measure the deformation field using something like particle tracking, but we didn't do that. Instead, since it's moving very fast and then moves right back, the incompressible assumption is a pretty good one. We can basically state what the deformation field is to pretty close order here. And then from there, given a certain position away, incompressibility will define the entire field, right? So if you have a position, you can't lose any volume, so that position has to translate during a bubble growth. So we can essentially just read out what the stretch is based on a radial coordinate. And finally, with all of those radii that we've defined in every one of our experiments, we could put numbers to what the stretch experience at each one of those different points was. And we know those are points of damage because we sort of stated that. From there, we can actually make these injury tolerance curves, sort of like the Wayne State tolerance curve, where it's like, well, you have this probability of sustaining damage. So and that's sort of how you can read this out. Like, these values down here are a 10% probability based on the whole population of what we've taken. So they're, with that bias included potentially. At 1.2 or so stretch, roughly a strain of 0.2, right? You have about a 10% likelihood of cell failure, or the projections being sort of ripped off, right? And so this is sort of like a conservative, like how conservative you want to be with your estimate of what it takes to break down cells. And that's sort of an interesting broader point of how conservative do you want to be to protect against, connectivity loss, for example. But we could certainly translate any of these values that we read off this plot to some strain it took to break down these cells. All right. So, there are, on this plot, there are 4 different curves, and they correspond to this, intracellular esterase activity, then all of the F-actin proteins that are here, then all of the microtubules that are inside neurons, and finally these dendritic projections containing MAP2 that are here. And they sort of look similar, but it actually seems to take less stretch to break down the dendrites that contain MAP2, which is, in itself interesting and suggests the existence of not just a strain criterion, but something that's size dependent, which would likely be a stress criterion. But like I said, we can really look at any of these, these thresholds. If we want to say our median amount, we can read off that value. If we want to say to your best, 10%, like 90% of cells should be okay to this tolerance, you can read out the 10%, for example. And they scale a little bit differently, right? There's this farther, expansion for the 10% for MAP2, for example. But the cool thing here is, we've got values. We can actually give this to our modeling colleagues, say, hey, this is what we really observed in a whole lot of tests, found that these projections break down with this, at these loading strain rates, which we can read off by, the same deformation field approach, and actually put some numbers to it. And really interestingly, there was a study by Ashfaq Adnan in 2018 that looked at MT microtubule dynamics simulations, which this is molecular dynamics, so it's going to be a lot faster rate than what we can really reliably achieve in the lab here. But actually, the failure strains are remarkably close to each other. So this what we sort of predicted as our value here of 0.14 for a median is right in line with the, I think, 0.16 from the maximum curves in his paper. So it suggests that like maybe after a certain strain rate, it doesn't really matter anymore, that you'll break it at some small strain. So that's sort of what I wanted to talk about today. I'll be happy to take questions, but maybe one last thing of just what sort of my group does and where we're taking this forward. I'm not still sort of doing brain work anymore, but we do characterization actually using this cavitation approach. And so we actually, I guess more broadly, use optical techniques to get usually full field deformations, determine properties inversely. But we use these same bubble dynamics with a theoretical numerical model, incorporating some viscoelastic effects, and actually a way to characterize materials inversely at these really high strain rates. So we try and get at this in an inverse way and really tackle some of these problems of like, well, you have mechanics, how do you get at assessing how the materials behave at these rates? And one sort of teaser here, we're actually currently building, this is my student Bashir, we're currently building this setup with multiple viewpoints where we can actually look at multiple perspectives of a bubble on the same sensor, look at asphericity, see what the deviations are, and try and get more about the field, and then connect that to material damage and such. So, we're still working the bugs out, especially thanks to COVID-19, but we're encouraged by the early results so far in reconstructing these bubbles in 3D, for example, which has been pretty cool so far. At any rate, that's, yeah, thank you for having me. I'm happy to take questions, continue the discussion, and thanks a bunch.
Thanks so much, John. That was awesome. Very cool work. I'm like thinking about all of the different pieces that had to come together and how hard it must have been to make sense of all of that data.
Oh man, it's especially like, and actually, I mean, that point is really good of just make sense of a lot of data is so underrated as like a skill set, especially for those of you in the crowd who are grad students. Spend a lot of time in your metadata. Spend a lot of time in that, like really constructing sensible processes and data management policies to get good metadata. So when you look at an experiment like I am from 10 years ago or something, you're like, oh yeah, of course, I know exactly what I was doing here. It really makes such a world of difference and would streamline a lot of processes, I'm sure.
Yeah. As sort of like, oh, go ahead. Manuel, if you have a question, please go for it.
Oh, no. I was just going to compliment John on the work. It looks amazing.
Thank you.
I don't know whether it's okay for me to kick off maybe with a very general question, but one that I would have if I was a student, or maybe didn't know yet to have, but should have. When you listen to a talk like yours, it's just amazing, right? I mean, the complexity and sort of the range of skills and tools that you're using is huge. And so I think as a grad student, I would look at this and be like, holy cow, this doesn't compare at all to the little project I'm working on. maybe I'm not, made for this world. Can you give us a sense for like what it took for you to put this together, right? Like how long did it take? What did you build it on? How many months, years did you struggle with this so that you have this like really shiny, beautiful 20-minute presentation at the end of it?
Yeah, absolutely. And thank you for the platform, even to discuss this, because it's something I feel really passionately about also. So I'm a first gen college. My dad's an immigrant. My parents have nothing to do with academia whatsoever. I mean, I had nothing to go on coming into grad school. I didn't know what the process would be like at all. And even now, I feel sort of constantly like when I figured out a problem a little bit Slumdog Millionaire here, that like I've just happened to find out the exact piece of information that I happened to need at the time, and it's sort of an accident all the time. Like, there's definitely this what's the term? Imposter syndrome. Thank you. Yes, there's definitely just this imposter syndrome that is very much rooted within, I think many of us still, even in what might appear to be, a nice shiny study on the outside. But what went into this was a lot of failure all the time. Really, like, there were days in the lab where I was just working on protocol days. There were weeks and months that I was working on protocol and was just babysitting a setup because I didn't know what a syringe pump was. Like I had many of these late nights that could have been made so much easier by just asking people and finding out, best process of things, and rather than trying to soldier through it. So for one, there's a network that you should tap into and rely on and ask your peers, because they might have seen it. They might know a thing or two because they've seen a thing or two, to quote a commercial. So that's one. The other is you sort of build the skills you need. And if you believe that, you have the potential to learn anything you need to, and just time might be the thing you spend to do that. Generally, with enough time and effort, you can get to places. And I think the PhD is really a mark of persistence and tenacity rather than brilliance. It really is just you spend time and work hard and do things and have people that advocate for you in your corner. And by those means, you get to the places that you want to be and answer interesting questions. But just keep at it. To those of you who are struggling with a process, like, I've been there too. All of us, I'm sure, have been there at the faculty level. We're just you guys fast forwarded. That's all it is. We're just you guys fast forwarded a few years. So keep at it. You'll get there.
Thank you, John. I really appreciate it. Word to the wise.
I'm happy to tell you guys about all the weird things that have gone wrong too. So
no, John, like. I feel like after that answer, it's like, I don't know, that was just a beautiful answer. And I'm like, my next question is like, okay, so.
And I'm sure you also would be willing to help folks that do similar research skip some of those mistakes you've made.
Yeah, are you kidding? Like, yeah, email me. My email is right up there. Feel free to
contact John.
Or tweet at me. Either way, yeah, no problem.
Yeah, maybe sort of like building off of your previous answer, could you talk a little bit about the timeline of this project and how long like each sort of, experimental and then data analysis and theoretical component took to kind of come together, and maybe some of the things that you were standing on when you made that happen?
Right. Great question. And actually, I should note that my PhD finished in 2017, and this is the sort of major publication out of part of it. So, it has been an ongoing process, and that's in no small nod to Harry Kramer, who's really taken especially the bio side of things in a really major direction. So tremendous hats off to Harry here. But the cavitation area, and maybe I'll start even earlier. I started my PhD in 2011, and I think we started originally looking at I guess more TBI broadly, and this was compressive studies. We have a paper from 2016, and a lot of that was building up protocols because our group hadn't done it before. Christian was advancing into a new direction, so developing those protocols took a couple of years, at least, like really getting those squared away. And this is like, I'm a mechanics student coming from a material science background, doing primary harvest of rat brain cells. I mean, this is, fish out of water a little bit too. So there's learning experiences that, are required there. And then moving into cavitation, getting the optical platform down probably, took another couple years, plus, the theoretical model, which was another year. So like, 5 years in, like, the pieces are now coming together of a lot of it. And even still, we didn't have all of the immunostaining. So that was then the next few years from Harry Kramer. Getting the 3D staining process took a lot of, challenging nights for Harry. And now we're finally there, but we're, it's a long study to try and get, especially this amount of data. I mean, it's hundreds of data points, lots of experiments, lots of volumes. I mean, yeah, it took a while though. Definitely took a while.
Yeah, that makes sense. I think we have a question from the audience from Carrie Dugan. Please excuse me if I'm saying your name wrong.
No, you're saying it right. Thank you. That was a great talk, John.
Thank you,
I've
Carrie.
been following your work for a while, so
I appreciate that.
to hear you talk about it. So as someone who also does TBI research and I focus on astrocytes, and I'm interested in looking at long term. So I'm curious with your system, if you can continue culturing past this cavitation state and look at some longer term responses that may be related to secondary, even if you're putting in other cells besides just neurons, whether that's being done or planned, or your thoughts.
Right. Great question. And that's yes, the natural extension to this primary injury study. And actually, the short answer is that it's in the works. I think actually Christian is paired with Diane Hoffman Kim over at Brown. And Harry, you could certainly jump in if you have more details on this, because I'm possibly out of the loop on what the collaboration is ongoing right now. But as far as I understand, you could certainly continue culturing these, and you can continue. I think actually, Diane has a spheroid model which shows a lot of promise in terms of like even developing protovasculature and these coupling between the oligodendrocytes, astrocytes, other progenitors, and neurons, of course. And then, yeah, I mean, it would be cavitating near them to try and understand, exactly what deformation we're applying to them, and seeing how they respond. But I mean, there's just so much. There's so much. There are so many directions that are to be explored. It's almost hard to pick one, right? Because depending on what you want to look at and what kind of behavior you want to pull out requires a different study parameters. So I mean, yeah, in the works, and I think it's just a huge open expanse of potential untapped direction. But yeah.
I know that's awesome. No, I appreciate your insight as someone who's also writing protocols, because it's not been done yet. I can really appreciate not knowing what direction to go because it's so wide open. But thank you.
Thank you. And Harry, by all means, feel free to weigh in.
Yeah, so I just wanted to jump in. So one thing to note about this model, that's really a nice strength of it, is that it is a co-culture. So we do have astrocytes. We have very little in the way of oligodendrocytes, and that's primarily a function of cell density. But we are, like John was saying, actively exploring the secondary injury pathways. And at the moment, we're sort of focusing more on broad strokes, sort of answering the hypothesis of whether or not we actually see a secondary injury response from these cells, as like the periphery of these samples experiences virtually no deformation. So I'm looking at simply at this point apoptosis. I'm adding in some GFAP staining to look at astrocytes specifically to see whether or not we're seeing an astroglial response. And that data is sort of just starting to roll in now. But yeah, it's definitely a wide open a field at this point, which is really, I mean, as a grad student, it's an exciting place to be because I can go any direction I want and still be publishable, as it were. But yeah, that's a great question.
Thanks, Harry.
Thank you, Carrie. And thanks, Harry.
Yeah. If there are additional audience questions, I encourage people to raise their hand or put something in the chat, and I'm happy to read it on your behalf. But I have one brief question, and then maybe we can see the lab video or lab Gathertown thing that you mentioned, John.
Oh yeah, sure.
My
Oh,
question.
yeah, go ahead. Go
I,
ahead.
yeah, sorry. I was wondering, have you sort of? I assume you have thought about this. So I guess it's sort of like, what are your thoughts on the relationship between the direction of the cavitating radius and then the projections on the cells? Yeah, I'm just curious as to the way that you all are thinking about that right now.
That's a fantastic, fantastic question. And so the decoupling of whether they're broken based on the direction they're pointing is the subject of a future study. And so we have a 2018 Nature Protocols paper, actually that one in the SciRep paper we've got. We looked at the segmentation of these neurons and tried to look at what kind of strain fields were being experienced by neurons based on the way they were pointing, right? Because they're these long spindly, creatures as they were. And right now, we haven't constructed these in such a way that we're fully segmenting the neurons. So like we don't have the capability with the data set we have presently, but the pipeline is there, which would then be like, can we actually, go by orientation and then see what the damage looks like in one versus another? Right now, we're sort of treating it as an equivalent strain. So, you could sort of do the Mises version and say like, well, maybe it's, to use plasticity terminology, maybe it's like J2 dependent. And that's our first shot, right? But you're totally right. I mean, there could be a directional dependence. We have, actually, I wonder if I have a backup on here. I mean, like neurons definitely behave differently in shear versus tension versus compression. I mean, different things will break. So it's tough to say because we have this deformation field that's a combined stretch in one direction and compression in two other ones. So which part of the strain field is doing the damage is even still an open question. But it's a really good one, and it's certainly an area to explore. But it'll be future work.
Yeah, for sure. I mean, the additional image analysis steps in that are definitely not trivial. I just had
Yeah.
to ask because I was like, there's no way they haven't thought about this.
Yeah, no, it's a really good question, and it's one that we sort of tossed around a little bit, but even just the first of like from a equivalent strain perspective, like what do we even have just if it's radial?
Yeah, and then Adrian, did you want to go ahead with the final question?
Yes, I have a very, very technical question. Amazing talk, amazing slides, everything. Super, super cool. And very technical question more on the mechanics side, because I know nothing really about cavitation on these hydrogels. How much control do you have? So if you change laser parameters, can you create different strain rates, different maximum radius? That's on one hand, and on the other hand is how much can you change the properties of the hydrogel and still culture these neurons, or is that just sort of like fixed? You have to use this hydrogel. You can't really change the mechanics because then you wouldn't be able to culture cells. Because I'm guessing you could either change the laser or change the gel in order to have, for example, different strain rates.
So I'll take your question in a few different parts. The first one about the first compliment on the slides, I'm going to deflect in part to Harry because some of these he's really been has constructed graphics for, and so I don't want to take full credit for all the slides. Harry did an excellent job. Your point about the laser settings, so we're able to change maximum radius straightforwardly. That's just an analog input on the laser. It's essentially a polarizer that you can rotate that just allows more energy in. That's a straightforward one. The strain rate actually gets faster when you have a smaller bubble. It's a little bit counterintuitive, but it's just that you're getting up to a maximum strain. Like the strain stays relatively consistent between different sizes of bubbles. So if you achieve that more quickly in a smaller bubble, you're actually getting to a higher strain rate faster. So we can control that a little bit. It'll range anywhere from the larger bubbles are probably closer to 10 to the 3 expansion. They collapse faster actually. And the smaller bubbles, it really scales actually linearly with radius. So if you have, around 300, maybe that's 10 to the 3, around 30 would be then 10 to the 4. So that does scale. So you can change the strain rate a little bit, but not to a tremendous degree. The mechanics of the gel though, we can modify it a little bit based on concentration. We didn't perturb that direction because we were looking less at the kinetics and more at the kinematics. So the kind of like the material properties, we determined what they were from the inverse, cavitation rheology technique. But it was sort of, we could use a different gel and would expect the behavior to be sort of the same because, we've seen it to be sort of kinematic. But that could be something we would do. We could try certainly, and just like increase the concentration of collagen, for example. These are 2.2 mg per mil, which are on the more compliant side, but we wouldn't expect the result to be any different, I guess. But that's certainly something we could try actually, and that could be a really cool, thing that Harry looks into.
I do want to jump in real quick because John has a tendency not to toot his own horn. The latter part of your question about whether you can change the gel, the really great thing about this technique is anything you can get a laser into and with sufficient energy to generate a bubble, you can perform this technique in. So the original work was designed at characterizing materials, and we extended it into the biological application. But basically, if you can culture cells in it, you can perform this kind of method with it, as long as you have the optical access to be able to image your bubble.
Cool.
Yeah.
Thanks.
All right.
Thanks, Adrian. Appreciate
Yeah.
it.
Thanks, Adrian, for the question, and John and Harry for the responses. John, can we still see what is it, like photos of your lab, or?
I'll show you. I'll show you, Gathertown. So
After,
I use
Emma really wants to see it apparently.
I don't know.
It's pretty fantastic. So I mean, it's.
Oh, so do I. I want to be very clear.
Yeah. I don't know. After John mentioned the Oculus Rift in class, I more just want to see how he's going to share it with us.
All right. So let's see if I can find my desktop that has it. Oh, here we go. Let's see. Oops. Give me 1 second. I just have to make sure my my screen share gets what I want to do.
Yeah, absolutely.
Can I ask a question to Harry in the meantime, because he also knows the technicalities? How is this laser bubble comparable to the cavitation event you would expect in an actual TBI? So, because I think the introduction was a lot about you need an interface to have this pressure drop versus just inducing it with a laser. Can you comment on that?
Yeah. So in techniques that generally revolve around the generation of these events with pressure, you tend to see the formation of clouds of bubbles that eventually coalesce into a single bubble, and then go about their general dynamics. With the laser-induced method, the big strength in it is that you do get a single bubble. So it's sort of hotly debated whether or not cavitation occurs as a direct result of pressure, like overpressures in the brain. It makes sense to us from a conceptual standpoint, and the modeling community seems to have a pretty good sense that they would suggest it happens. But within the medical community, it's one of those, we can't see it, so how do you know it's happening? And there's a skull in the way, so we can't hear it. But the general idea would be that we would probably expect sort of clouds of bubbles to form rather than single bubbles, but the governing dynamics should remain unchanged. Even in the laser-induced case, once you've reached maximum bubble radius, it's a pretty good assumption to say that you've reached thermodynamic equilibrium with the surroundings, and so your collapse and subsequent oscillations are all driven by the same governing physics you would expect to see in situ.
To piggyback on that, actually, there's quite a sizable histotripsy, like a tissue breakup using ultrasound group here at Michigan. And they've run ultrasound versions of the same sort of experiment of cavitation, like inducing cavitation with ultrasound instead of laser, and the dynamics are remarkably similar. Like really, the growth process, although different, you have a different pathway of the energy to be absorbed, back into the sample because you generate plasma for laser. Actually doesn't really affect the resulting dynamics very much, like for ultrasound versus laser. In fact, there's work on an ultrasound cavitation rheometry technique, which is a pair to the laser cavitation rheometry that we had developed originally. So pretty yeah, translatable actually.
Thank you, and sorry for taking away from
No, not at all.
Gather Town.
All right, check it out. This is my lab. So welcome to the lab. This is pretty much, you know, the space we work in. And so I have these little pictures of what the space is. So this is all of Bashir set up here with, actually, I think, yeah, I can zoom in. You guys can see this, right? So this is on the table. We've got, you know, all of the optics. This is our high-speed camera. It's a Shimazu HPBX2. This is our laser with the power supply underneath. This is the power supply for the camera. And all of this is to generate cavitation inside materials. And let's see, I can leave that space and we can look at the other side. So inside here is the multiphoton microscope that we got for our group. There are some boxes with unknown things in them, which is really great. But yeah, this is sort of the space we've got. It's a lot of, you know, fabricate soft materials. We have, you know, some room and in a different space for some bio culture, for example. Then here I have a teleporter, which I wish I had in, you know, in real life to my office. So this is where I'm sitting right now. You can see the door that's over at the top of the room there. I've got my espresso machine in the virtual space. And then this is the actual bigger lab that we have shared space in with a whole bunch of instrons and a drop tower. There's a DMA over here. Let's see, over here is the DMA. We got one of these. Yeah, so then, so this is part of Ellen's group. I share space with Ellen Aruda, and so she has her split Hopkinson bar set up over here. Yeah, I mean, but it's a nice space. There's a lot of room. There are 4 of us that share the bigger area, and a lot of good stuff we could do. Let's see, over here. Is this the one I want? No. Yeah, so this is over here. There's sort of bio stuff. There's like a small tensiometer for bio samples. And then I even made the, so I actually made this space because I use GatherTown for like my office hours for my class, and it's amazing because we're doing remote office hours this semester. So this is where my grad students sit in the office. They have their little spaces. And then, you know, because I wish that we could have better things, I made like a lounge area so we could play games if we want to.
Well, I'm naturally curious now where the question marks lead.
Yep, gotta find out where the question marks are. Turns out it's going to go out to a seaside.
Wow, maybe I should look for a job in Michigan.
Yeah, if only we actually had this. This is just me living out my wildest dreams virtually.
That's fantastic.
But anyway, yeah, that's our virtual space. I'm hoping to, you know, if anyone wants to virtually visit at any point, you know, I'd be able to show the space and we can show results and posters and whatnot. So if you want to virtually visit Michigan, just let me know.
That's awesome.
I'm not gonna lie, you set the bar pretty high for this season's journal club. You know,
Good.
We got videos, we got animations, we got Gathered Town. We got interaction with additional folks. Yeah.
Next time I'm back, I'll make sure to get the VR incorporated.
next journal.
Next time, yeah. Well, thanks, guys. I appreciate you guys hosting me. I'm deeply indebted to those of you who ask questions because it's definitely got my brains, you know, working. But yeah, thanks again.
Yeah. Well, thanks so much, John. And thank you for everyone for coming and listening. This has been awesome. And we have another speaker next Wednesday. I actually can't remember off the top of my head who the next speaker is. Maybe Manuel, do you have my back here?
I'm pretty sure it's Kiara, but I gotta be honest, I'm looking it up right now.
All right, watch Twitter, check your emails.
Yeah,
our next speaker will also be awesome.
Next week is Chiara Bellini from Northeastern. So we're very excited about that.
Yeah. All right. Well, thank you, everybody.
Thank you. Thanks, John. Phenomenal.
Thanks, everybody, for being here.
Thank you, guys.
Thanks, John.